Good afternoon, and thanks everybody for showing up to this event, Zoom event today. So my presentation will talk about some of the collaborative research I've been doing on bats that's related to the COVID-19 pandemic. I'm not sure whether my inset video image of myself is showing up, but I've got a background photo of the bat cave at the ROM. So during the current lockdown, this is one of the obvious impacts on the public side of the museum is that, the museum is closed. So we can't see the exhibitions or galleries at the ROM, but we're trying to keep up with research, with the research aspect as we're talking about today. So the picture on the top is a species of horseshoe bat, which is the genus implicated as a natural reservoir of SARS-like coronaviruses. The image below is of the spherical SARS-CoV-2. So this is one of two severe acute respiratory syndrome viruses that is responsible for the highly contagious and deadly disease that has caused the current global pandemic that we're in. As you can see, the virus is surrounded by numerous spike proteins, which bind to the ACE receptor sites on the human cell that you see on the bottom there. And this is how the virus enters the body and then uses the host to replicate more of itself and cause infection. So bats don't transmit SARS-like coronaviruses directly to humans, but there is a connection between bats and coronaviruses. However, there has been some false information circulating around, especially on social media. One of the most infamous was the suggestion that the outbreak got started by people eating bat soup. But this was based on an old video on YouTube that predated the pandemic. Unfortunately, this led to the wrongful persecution of bats, including killing them in several countries throughout the world. A study by Liu and colleagues published in the scientific journal Lancet, and other studies as well, have found that the closest known coronavirus to SARS-CoV-2 occurs in a species of horseshoe bat from China. However, there is a 4% difference in RNA between the bat virus and the human virus. So to give you an idea of what that means, chimpanzees and humans are about 5% different in their genomes. So 4% is actually quite a bit different as far as genomic variation goes. Calculations were also published by Bonnie that go back in time to estimate that the most recent common ancestor of the two viruses, of when the most recent common ancestor of the two viruses existed. So this actually dated back to 1969, as you see in that left inset down below. Which means that the virus has been circulating unnoticed for the past 5 decades in some other unknown intermediate host before jumping to humans. And then in this last example here, so another difference is found in the coronavirus spike protein. So you can see, I'm not sure if you can see my cursor, but this red part is the spike protein on the upper diagram. So this is what binds to the ACE2 receptor in humans to gain access into cells and infect the host. However, the bat virus, which is the sort of second line down on this A figure, so if you can see my cursor, you can see I've also marked it off in red on with the text bat and also the arrow. But there are 5 amino acid residues that are different from the human virus. Which is on the top line of that figure. So what this means is that it makes it more difficult for the bat coronavirus to infect humans directly. So these are important pieces of information that tell us that the coronaviruses in bats and humans are similar, but different enough that transmission was through a still unknown intermediate host, so not directly from bats. Okay, so that gives us a bit of background information on the association between bats and coronaviruses. Now I'll get into the connection between the ROM and COVID-related research. In 1989, we began preserving tissue samples for genetic study, along with a voucher specimen for research, primarily on the evolution and biodiversity of mammals, but also for the discovery of several new species to science. Over the last 3 decades, we've collected almost 15,000 bat specimens, so not anywhere near the millions that Sebastian has in his invert collection. But we've collected those specimens from about 30 countries throughout the world, and those are the ones that are highlighted in green on the map over there. Most of the field work has been in tropical rainforest areas of South America and Southeast Asia, where species diversity is in general the highest in the world. And then the picture below is of a 2015 field trip to Sri Lanka, where we were exploring one of the last remaining remnants of old growth forest in the country there. So during these 30 years of field work, the bat frozen tissue collection at the ROM has become an important resource and one of the best in the world. It has grown to represent 15 of the 21 species of bats, 120 of the 220 genera, and about 400 of the 1,400 species of bats. So the photos on the slide give you an idea of the range of morphological diversity in bats, from the white stripes on the back of this 2-line sac-wing bat to the white stripes on the face of the Trinidad big-eyed bat on the left here, and the big ears on the royal horseshoe bat that's on the right, photograph in the bottom there. So in addition to having good geographic coverage and good taxonomic coverage, most of our tissue samples were originally quick frozen in the field in liquid nitrogen, which is really cold at minus 196 degrees Celsius. This is important in that the usual method is to preserve tissue in ethanol, but this makes it difficult to recover long fragments of DNA for some of the newer technologies of whole genome sequencing, which I'll be talking about in a second, but also more difficult for the study of viruses, which have RNA instead of DNA, which is less stable. In addition, getting liquid nitrogen while we're doing fieldwork can be a bit of a challenge at times, and also lugging around this tank in remote areas is also a bit challenging as well. But this is still the best way to preserve tissue for molecular studies, especially the newer methods, and also for long-term storage. Because basically, the colder you keep it, the less the DNA degrades over time. Our tissue collections are here, to study, right now, but hopefully they'll be around for, centuries and centuries and generations down the road for further study as well. Okay, so the last part of my presentation, I'll be concentrating on some of the collaborative projects that are related to pandemic research and bats that I'm doing. So just quickly, the first project looks at bats and sequencing their genomes. The second one takes a perspective from the viruses and about developing vaccines. And the third is a preliminary discussion on establishing a joint lab and field program on emerging viral pathogens in Laos and Southeast Asia. So I've sort of broadened my presentation beyond the original title that I had presented to the pandemic research event. Okay, so for the first project, which is on bat genomes, so this is part of the BAT1K consortium of researchers from around the world. The objectives of this initiative are separated into 3 phases. So phase 1 is a sequence of species of bat in each of the 21 families of bats. Phase 2 is a species in the 220 genera. And then phase 3 is getting the genomes of all the remaining, 1,400 plus species of bats. So currently we are in phase 1, and the ROM has contributed frozen tissue samples for 1/3 of these bat families. So these are the bats that have pictures of their faces. So again, you see there's quite a bit of morphological diversity within these 7 different families of bats. So BAT1K involves many universities and museums, but the main DNA sequencing collaboration is with the Max Planck Institute in Germany. So that's their logo down the bottom left. With the outbreak of the pandemic last spring, the Max Planck Institute got funding from the European Union to sequence the genomes of species in the horseshoe bat family Rhinolophidae. So that's the bat pictured in the bottom there, which is considered the natural reservoir of SARS-like coronaviruses, and also its sister family, the old world leaf-nosed bats, Hippocidaridae, which is the picture sort of in the middle of that bat. So a bit of background on viruses. There are 4 types or genera of coronaviruses. Alpha and beta coronaviruses are found in mammals, whereas gamma and delta coronaviruses are found in birds. So illustrated here are the bat coronaviruses. Yeah, so this is actually, I got this from this publication here in Nature Communications. So this is just the B part, the beta coronavirus part. So within the beta coronaviruses, they're separated into 4 subgenera, which are what you see on the right-hand side. And that, excuse me, and that includes the sarbecovirus in blue at the bottom of this tree diagram. These viruses occur in horseshoe bats and some other mammals. 2 of these sarbecoviruses have caused the SARS outbreak in 2003 and the current COVID-19 pandemic. Up on top in purple are the morphological and these include beta coronaviruses that caused the MERS or camel flu outbreak in 2012, which is a less contagious disease that was transmitted to humans from camels, with bats as a suspected reservoir. So overall, this study by Latine also suggests that the horseshoe bats in the family Rhinolophidae is the ancestral host for the beta coronaviruses. So you can see that, again, if you can see my cursor on that diagram, that the blue branch and Rhinolophidae goes to the base of these beta coronaviruses. So they're implying that the ancestral beta coronavirus that includes the SARS-like coronaviruses, they're suggesting that the host is these horseshoe bats in this family. Okay, and as a part of our second collaboration with the BAT1K consortium, we sent frozen tissue samples for those 8 species of horseshoe bats and also old world leaf-nosed bats to the cell biology and genetics lab in Germany for the COVID-19 study. So this will involve sequencing the genomes, assembling the 2 billion DNA base pairs. So 2 billion obviously is a huge number. So in comparison, the typical beta coronavirus genome is only about I think 20 or 30,000 base pairs, not not billions. And then the final step is annotating these genes. So from there, we can look more closely to identify and compare parts of the genome, such as the ACE2 receptor sites, to see how similar or different they are between bats and humans. Also, what genes are involved in the bat immune system. These comparisons will give us a better understanding of why bats have a lot of coronaviruses, but typically they're asymptomatic and they actually appear healthy. So I think a lot of the clues to sort of solving, the human health medical issues, because, when, people get, these SARS-like viruses, they're actually quite deadly. So some of the answers might lie with how bats deal with these coronaviruses. Okay, so the second collaborative project that we're working on is on the virus vaccine. And this is in collaboration with virologists at Western University. So when the COVID-19 pandemic hit last year, they were just starting clinical trials for a vaccine against the MERS coronavirus that I just talked about earlier. But then they quickly switched their clinical vaccine trials to the more pressing concern of the SARS-CoV-2 problem that was occurring. So their sort of vaccine method involves inserting the coronavirus spike protein gene into a replicating viral vector vaccine, as outlined in this schematic diagram on the right here. So basically, the viral vector develops an outer layer that looks like SARS-CoV-2, which initiates an immune reaction from both the spike proteins in the vaccine, but also this viral vector enters the cell and also replicates more spike proteins. So that generates an even stronger antibody response. So these new clinical trials started last October with favorable results. But with others, getting an earlier start on the COVID-19 vaccine, such as Moderna and Johnson & Johnson and Pfizer, Western has refocused their efforts on using their replicating viral vector system to produce vaccines for potential outbreaks in the future from other coronaviruses. So that's when they contacted us in November to ask us about our bat frozen tissue collection from Asia that a mutual colleague of ours told them about. So we've been down doing Southeast Asian fieldwork since 1993, and more specifically from 2001 to 2007 in China, where the two SARS-like diseases originated. So here's a picture of me in one of the caves we surveyed with mist nets in 2006 in China. So the virologists from Western came to the museum in December to directly subsample from our frozen tissue collections. So this included 211 specimens representing 56 species of bats from 4 provinces in China, which are highlighted on the map in the upper right of this slide here. So in February, a joint media release by Western and the ROM was sent out highlighting the collaboration of our 2 institutions on developing coronavirus vaccines. It got picked up by several news outlets across Canada and also internationally. So far what we've done is the tissue samples have been screened for coronaviruses and the RNAs have been extracted from those tissue samples. The spike proteins are now being isolated and amplified for sequencing. So the preliminary results are encouraging, and they actually hope to come back to the ROM sometime next month to broaden their sampling geographically to Southeast Asia, so beyond just China, and taxonomically to other small mammals beyond bats, such as rodents and shrews, to get a better idea of how widespread and common coronaviruses are in species of mammals. And finally, the third project that we're collaborating on that I want to quickly talk about is actually a very recent development that just happened over the last couple of weeks. I was contacted by Global Affairs Canada to join with several other Canadian institutions, including universities and federal agencies, on an international partnership with Laos and the Pasteur Institute. So they wanted to discuss the possibility of establishing a joint lab and field program studying emerging viral pathogens, including new coronaviruses. So this joint lab setup would include a field component doing biodiversity surveys in Laos, a lab component also in Laos to identify zoonotic diseases, and the establishment of a pathogen database for further study. And we actually have a previous connection to Laos. So there's a ROM research associate that has collected approximately 74 species of bats from throughout the country in the 1990s. So that's pictured in this map on the right. So you can sort of see Laos is sort of the main image here. You can see all these little pins are the different localities that the ROM research associate surveyed back in the 1990s. So it covered most of the country. So in addition to other field work that we've done in Southeast Asia, so now we have actually a good foundation of sort of bat species diversity in Laos, and we can basically already contribute to this collaborative project. And in fact, we have a meeting later tonight to meet with our international partners in Laos, where of course for them it would be the next morning for them. So hopefully we'll have some more positive news to report on this endeavor soon. So just to quickly summarize what I've gone over here. So some of it might have been a little bit technical, but there are several sort of take-home messages that I want to get across. First is that bats don't transmit SARS-like coronaviruses directly to humans. It goes through an intermediate host that was originally thought to be a pangolin, but even the spike proteins of their viruses are different enough from the human virus that pangolins probably didn't cause the infection that led to the current pandemic. So we still don't know, what was, the immediate transmission host that caused COVID-19. Secondly, encroachment on wildlife and their natural habitats increases the chance of viruses jumping between species and mutating before infecting people. So animals, aren't at fault, but in fact, they are actually beneficial to the ecosystem as seed dispersers and flower pollinators, such as this nectar-feeding bat that's pictured here. Another point is that bats have a lot of viruses, but they are typically asymptomatic and appear healthy. So studying bats and their immune systems will help us in controlling any new viruses or diseases that emerge. And finally, developing a repository of ready-made vaccines for different types of coronaviruses will give us a head start on any new outbreaks that might happen in the future. Okay, so thank you for listening, and I hope you got a better understanding of bats and how they can help in medical research. And now I will pass it on to Alex to talk about the pandemic and masks.